Pharmaceutical Processing, in conjunction with INTERPHEX, presents INTERPHEX Live.
So if you've been following the panel discussions through the last couple of days, you'll notice that every time we got the CAR T on there, it doesn't matter if we're talking about building stuff or developing, we packed the house. I guess there's a little bit of curiosity. So I'm glad we have assembled a group of experts. Okay. So I'm going to stumble over everybody's name and I'll get the introductions. So we have Boro, Dropulic, that's Miltenyi Bio, is that right?
Boro, Dropulic.
Okay. And
Lentigen, which is a Miltenyi Biotech
good.
company.
All right. And we have Mohamed from CMC Immatics. I got that one right. Okay.
Close.
Yeah, close is good enough. And then we had Heidi Hagen from Vineti, is that right? Yeah, there you go. And Peter Olagunju, is that right?
Close. Olagunju.
Okay, and that's from Bluebird. I got that right.
You got that right.
Okay, and Katie Worrilow, from founder of LifeAire Systems. So, what we're going to talk about is we're going to go around, let everybody know what each individual person's got a skin in the game here. So they're going to each give a little bit of background. So we could start. Seeing that I blew your name the worst, Boro, so you start.
No problem. I've been in the gene therapy space since the late 1980s. I was at the NIH, then at Johns Hopkins, and then I spun out my first company. We were the first group to put lentiviral vectors into the clinic and show that there was safety with our clinical collaborators at the University of Pennsylvania. Then I started my second company, Lentigen, and we are developing lentiviral vectors. We were acquired by Miltenyi back in 2014 with the idea of integrating what we do well, lentiviral vector manufacturing, with automated device cell processing, which is Miltenyi Biotech's specialty. And what we do is we enable clinical sites and companies with lentiviral vector manufacturing and with automated cell processing, and then we also have a clinical pipeline of our own.
Great. Good afternoon. Peter Olagunju. I have been with Bluebird Bio for almost 4 years now. Bluebird Bio is a company that focuses on autologous therapies, personalized therapies for rare diseases, mostly blood disorders, and then also oncology. At Bluebird Bio, I lead a group called Patient Operations, and it's within the CMC function, so your chemistry, manufacturing, and controls area. But it's really kind of what we call the tailored fit of how we organize to support the fact that the manufacturing process begins with the patient's cells. So really organizing your CMC operations around that. Prior to Bluebird, I spent a little over 5 years at Dendreon and worked there with another panelist who will probably mention that here in a bit. But yeah, just really excited about what's ahead for the cell and gene therapy industry and the opportunity for the conversation today.
Katie Worrilow, LifeAire Systems. As a clinical provider and practitioner of personalized medicine specific to the human embryo, we're here to share a great deal, 10, 15 years of extensive research on the molecular epidemiology involved with airborne pathogens specific to the living cell and the cytotoxic effects that it can exert on those cells. Despite working within a clean room. So our data comes from culturing the living cell in an ISO 5 clean room and how that clean room failed us over a 10-year period, what we were not considering. You don't know what you don't know. So we're here to share that information specific to not only the critical role of viables in your living cell environment, but also your volatile organic compounds and the levels that are necessary, really the airborne metrics necessary to support the living cell. And so I have a very cellular focus, and with a successful cellular focus comes a successful delivery to the patients.
Good afternoon. My name is Heidi Hagen. I am a co-founder and chief strategy officer of Vineti. I've spent the last 30 years in the biotech industry, the last 20 of that in cell therapy. First 10 of that 20, I was the Senior Vice President of Operations for Dendreon, responsible for bringing forth the first active cellular immunotherapy that was approved in Europe in 2012 and the US in 2010. responsible for all of the CMC and technical operations, including building the facilities and clean rooms, which we just discussed. Since then, I was actually a global COO for Sodio. It's a small cell therapy company coming out of the Czech Republic. Built a facility in China, did the operations in China, Europe, and the US, and opened up the Boston office. Since then, I have been a consultant in the industry. Half my clients are VCs and investors who are putting a lot of money into cell therapy. And I do due diligence, usually coming out of an academic center. Sometimes it's out in the commercial world. The other half of my clients have been cell therapy companies, typically anywhere from phase 1 to phase 3, looking for assistance in the operations, regulatory, and financial side. It's with that GE Ventures came to me and said, this is a very burgeoning area. Back when once upon a time, Peter, who discussed earlier, we had to put up our own cloud, our own system that was an IT system that allowed you to order these products at the medical center, do a scheduling, cell collection, transportation, manufacturing, and then administration and reimbursement triggers after that. So what we do now is we're providing that to the industry in whole with a cloud-based system that does everything from order entry to fulfillment of the product and reimbursement, and it integrates all the software stacks between. So I'm kind of actually now doing the support on the side, on the software side, in addition to also helping CMC and operations folks.
My name is Ali Mohammed. I'm the Vice President of CMC at Immatics. It's a clinical stage company that is focused on TCR-based cell therapies. The TCRs are slightly different from CARs. They're in the same category, but they're more effective in solid tumors because they reach the intercellular compartment of the cells, unlike the CARs that they usually on the surface. Before Immatics, I have been in this field of cell therapy or cell and gene therapy for close to 20 years. Before that, I worked at a couple of CMOs. One of them is BCT, which became Hitachi in Allendale, New Jersey, and the other one is Lonza in Maryland. And in both these roles, we helped other clients, including Peter's company and others, manufacture their cell therapy products. So as CMO, we provided that service for process development and GMP manufacturing of various different types of cells for mostly oncology, but as Peter said, rare diseases and some other regenerative medicine applications.
Okay. Oh, I'm a big boy. They give me my own. So. Anyway, what I strongly suggest, considering the crowd, considering the vast amount of knowledge we have here, I'm going to try to move through this stuff pretty quick. But I strongly suggest you accost our speakers when they are done. All I ask is that you move a little further out so we can get onto the next session, okay? But there's going to be a lot of questions, obviously. And from what I can hear, there isn't anybody here who cannot answer it. So let's start with the basics. Peter, so obviously there's a lot of excitement here. You want to, so as a new class of medication, I mean, let's talk about it. Why is everybody jumping on board and why are we excited?
Yeah, so I think the excitement is around the data. When you think about CAR-Ts very specifically, there are two now commercially approved CAR-Ts, Yescarta and Kymriah, in an oncology indication, and that data in that indication has shown versus the standard of care just a really significant impact, positive impact to patients. So it's really around that data and offering patients another option. And then, as Ali mentioned, it's about what's ahead. So currently right now it's in CD19 or blood cancers where CAR-T is effective because it's targeting the surface of a cancer cell. But moving forward, really the leap that we're looking to make is the potential of going into solid tumors. So being able to unlock that ability to have an effective treatment, either CAR-T or TCR approach for solid tumors. And so that's really what is generating the excitement, is this idea of personalized therapies that have a long sustained impact for cancer patients mostly.
Okay. So anybody else want to share a view on it? Heidi, please.
I would also add to that very good answer is that we're getting more precise and more personalized in terms of both diagnosing and addressing everybody's individual needs. And things like that are exciting coming along with a lot of the cell therapies are things like neoantigens, where we're able to now do single cell sequencing, look specifically at what's going on in a particular cancer, and then address that from a personal perspective, both from an ex vivo cell perspective, and then eventually we hope that what this does is provide a platform of knowledge and capabilities of doing in vivo work. But we have to take our scientific and medical steps first, and I think CAR-Ts have provided that foundation and those first steps that'll take us closer to that.
Okay.
Yeah, may I just add, I agree with Peter, it's the data that's exciting, but also the rational way that these novel generation of therapies will be generated. Drug therapy is like going into the hills and hunting for gold, and hopefully you hit on something, and drugs could have a problem, very late stage preclinical, while these vectors, it's a very rational way that you design. It's more like an engineering problem. And so I think the promise is that we can build modularly on what we know and iteratively improve and have a high predictability for things that will work in late stage clinical trials.
All right, that worked really well. So Peter, I'm going to come back to you, unless somebody else wanted to add in. I mean, go right ahead.
So the field of CAR-T or TCR T-cell therapy is new, but it took actually many years to put it together. There is cell therapy, everybody knows about using cells for regenerative medicine, and also gene therapy when you use AAV or something to correct some genetic disease. However, we combine the two together by genetically modifying these T cells with a specific cancer antigen, either in the form of a CAR or in the form of TCR, to make live drugs, live medicines. Most of it at this time is autologous, where you manufacture one batch for the same patient from the same patient's material. So it goes one-to-one, and that's why it's challenging and it's expensive. However, oftentimes for these patients, it's their last line of therapy. They don't have any other place to go. Most of the industry, like Peter said, we have 2 CARs approved. We have the TCRs. Most companies have TCRs. Lagging a little bit behind, and this is one of the things I'm working on. However, the allogeneic field, where you can take cells from healthy donor, genetically modify them, or gene edit them to have them suitable for many other patients. That way you can have healthy donor with good cells, where you can engineer them to treat cancer for 100, 1,000, or 1,000,000 different patients.
Right, that was one of the things that came up in several of our programs. So thanks for bringing that. That's actually the segue to the question I was going to ask Peter. What are the challenges between the two?
Yeah, it's a difficult biology challenge, to be quite honest. So the idea of being able to have one donor material supply a drug that is applicable for multiple individuals is a great vision, and that's where the industry is trying to get to with companies like Allogene or Selectus and others. The biology is difficult in that the reason why we focus on autologous therapies is due to the hurdle when it comes to GVHD. So you want to start with material that will be recognized by the patient and not be recognized as foreign. And that's really HLA typing. And so to be able to solve that, you really have to go in and edit those genes that are specific to the individual, and then actually edit in genes again to make it more broad. So it's like almost double editing that has to take place to have an allo approach that would be broadly applicable. So that biology is really difficult. There have been companies that, in preclinical models and now soon to be entering the clinic, have shown kind of advances towards being able to have an allo approach. But you'll see that there's still many of the approaches are autologous approaches.
Yeah, one of the things, like I said, we've had several of these conferences. So my next question is really nested in what puzzled me is everybody thinks we're still thinking small molecule, we're still thinking biologic, we're not thinking cell, right? So I think in a lot of cases, what my point is, we're thinking our existing manufacturing infrastructure is going to be a one-size-fits-all, drop it in, and we rock and roll. And I don't see that as happening. It's based on what I'm hearing, there's a lot of ifs. So let's start talking about some things that doesn't make a one-size-fits-all. So why don't we start with Katie this time?
So we learned the hard way that one size did not fit all. It was 10 to 15 years of, as I shared a little bit earlier, very painful cellular failure. That cellular failure meant we failed our patients in the culture of our living cells in an ISO 5 clean room. We met and exceeded all GMP, FDA, and ISO 5 guidelines. We colored with inside the lines, and again, you don't know what you don't know. But when that clean room failed us was when it was failing to comprehensively remediate the chemical airborne pathogens, or your volatile organic compounds. And so what the failure of that clean room taught us was the airborne metrics beyond viables and non-viables that we had to control for to have consistent and successful cell culture. We deemed it completely unfair to our patients and their cells that they entrusted us with to have something that we couldn't even see or smell impact our living cell culture success. And so where did those VOCs come from? Everywhere. Every detail matters. The caulk in the fabric of your clean room, your pod, whatever you're using. I mean, the outside air. You will never control your neighbors and their actions. So our cells would suffer as a consequence of the hospital generator running on a Tuesday afternoon. Our cells would suffer when the medevac pad was resurfaced with the asphalt and the toluene. We have 10 years of coincident data showing outside events, or frankly, our own SOPs that were causing an elevation in the cytotoxic VOCs. And what this launched was a study of gas phase VOCs, so gas phase IPA. We all use IPA. Gas phase IPA, let's say you have 1,000 ppb in the air. What does that mean? How much of that enters your cell culture media and how quickly? And it's very quick. It's very hydrophilic. And then once it's in your cell culture media, in how many minutes does it enter your cell? And what levels of IPA in the cell are cytotoxic? And all of these learnings came from our negative data and the cellular failure of the clean room.
So Heidi, I mean, in your consulting business, you must have come across this a lot.
Yes, actually, and it's, as you think about it, living, breathing organisms rather than well-characterized molecules. And so in addition to, I think, what we discussed very articulately here in terms of the infrastructure, the air, and the manufacturing facility, you also have the fact that you have patient-to-patient variability for autologous. And with that comes a different starting material with a whole set of other variables that come in with it, and being able to separate the variables that are patient-specific versus those that have to do with the science and the process is really a very big data-rich exercise. However, something that we, I think as an industry, weren't prepared for. I was at FDA's standard committee body meeting last week on cell therapy and gene therapy. And it's interesting as you ask the regulators, and they know one size does not fit all for this area. This is a very, very important topic for our regulators and very important topic for us and our patients. However, I think we're still trying to catch up with the proper technology, and some of it's starting to come on board. Additionally, we also, I would say, in the bigger, broader data management area, most of you probably have heard chain of identity, chain of custody. So in the example of autologous cell therapies, what you take out of one patient better go back into that patient. And as you can envision, no longer does the supply chain live just inside the four walls of manufacturing. It actually starts with the healthcare provider, the patient themselves, where the cells are collected, all of the data associated with that, the environment around that data and around those cells, transportation, all of the manufacturing information, which I would also say there's one other aspect that we haven't discussed yet is analytics. So from a QC analytical standpoint, we're used to doing in the pharma business large lots, where for 10,000 doses, you have a set of QC methods, you do them, you have a C of A, and from there you go on. Now you're talking about one-to-one, a batch record that can be this thick if it's not electronic. You're talking about QC methods from the very start of it, which is outside of your four walls of manufacturing. And all of that information that comes through is critical to the success and quality and safety of that patient. And so I think we also were caught off a little off guard technically from a software perspective. We all walk around with these guys in our hands. We can do many things with this, but when it comes to patient healthcare records and it comes to data across all of those different parts and those geographies, we weren't prepared for that as an industry. And I think the FDA is very, very clear about the fact that they have an expectation, rightfully so, and so do the patients, that we collect that data, integrate it, and understand the complete patient journey from start to end. And that end, by the way, is not at the point of administration. It's actually well after administration. So there's a standard for the CAR Ts right now to follow it for 15 years, and that was just a deadline from a regulatory perspective. But I think realistically, when we put gene-modified, gene-edited cells into a human, we want to know what happens eventually. So I would say we're also talking about data management, data infrastructure, in addition to, I'd say, the physical infrastructure and the air quality aspects that are very critical to these unique products.
So Peter, I mean, we talked about the uniqueness of using patient cells, and there's a lot of considerations. I mean, and in our industry, including you, I mean, but the bottom line is, I got it. We think life cycle is life cycle. Sounds to me like life cycle is about to be redefined. Sounds to me like we're getting unique considerations on using patient cell. I mean, what's going on? I mean, I think we got to anticipate.
Yeah, I think the key point there is what Heidi mentioned, the patient-to-patient variability that's seen. And if you're talking oncology, maybe there's more numbers, but if you're talking rare disease, you're working with low numbers of patients. So to really be able to extrapolate data and take action from that data, you want a robust data set. Without that robust data set, you then have to create a manufacturing process that has enough latitude for that variability from patient to patient. Other measures that are taken are really organizing around that point of collection. So that whole apheresis procedure, which is the collection of the patient's cells, and how do you ensure the proper training, the proper procedures are there so that you can repeatably collect those cells so that material is suitable for manufacturing. So that's a technique that a number of companies are taking, is really organizing at that point of care, that collection of those incoming cells before it goes into manufacturing.
So, Katie, it seems, I mean, how does all this play into, so we went from how unique the raw material is, how the starting material, and, where we're thinking we're going to be able to do this ain't fitting what we want to do. So is there something else about unique that I'm missing, or?
Well, I wanted to follow up on a point that Peter made.
Right.
We were asked to do site assessments and environmental assessment of a number of leading cell and gene therapy facilities. And in doing so, we were able to witness and watch some of their SOPs. And a lot of the bulk manufacturing requires a certain set of SOPs, obviously, to remove your viables. And what we saw is that now that they're working with individual patient cells, in particular compromised patient cells, and the environment is so much more critical than in your bulk manufacturing world, a lot of those, they're using the same SOPs. And there's actually, what we had to learn as providers was we needed the same SOPs to reduce our viables, but there are ways that you can adjust those SOPs to also meet the needs of your cells. Because the way the SOPs are being performed now, the frequency, the type of sporocidals, the type of IPA, how they're being performed, the frequency of being performed, how they prep their material, how the personnel are prepped, seeing the intensity of that for the goal of no viables can be adjusted to better meet the needs of your cells and not add to the cytotoxicity of the environment.
So let me see if I got it straight now. So if all CAR T cell processing is going to occur in a clean room space, right, which is nirvana, right? We're talking from our end, our side of it, right? And so what I'm hearing now is that the environment still poses a threat to the live cell and the delivery of those cellular products to the patient. Did I get that one right? I mean, yeah, right? So, I mean, if that's the case, I mean, as an industry, everybody's jumping on a bandwagon, how do we start to work our way, how do we control that? I mean, do we have requirements, we have specs? Katie, you want to take a shot? Heidi, Did I summarize it right? I mean,
Yes.
all right.
I mean, I would say, again, we didn't know what we didn't know. And now that this information and data is available, is to expand the metrics that are required in the clean room. Just go beyond your viables and non-viables. And actually, if those metrics are achieved, there's a statistically significant increase in the cellular metrics that result from that environment, as well as the delivery of a successful product to the patient. So I think the first goal is to expand those metrics and then absolutely make sure you're delivering those metrics to that space consistently with challenges, with environmental events that are going to happen. So make sure you're meeting those metrics and can deliver them under all circumstances to protect the cells.
And I'll just tag on to that because I think it's acquiring the knowledge first and gathering that data is really critical. But then it's taking that data, aggregating it in such a way that we can develop new standards. So we've had standards in place for small molecules, recombinant proteins, and they work. But once upon a time, we did the same thing there. We gathered new data, looked at what was needed for the clean rooms, really established the ISO standards that exist today from those types of molecules. But now I think we're in a new age here where to serve these products, we are going to need to take that data and put it into some standard form and start to have maybe a different type of standard in terms of the products you have in your HVAC systems and in your air systems. And it might not be what you've used for previous facilities. It's probably a new standard coming along.
So Heidi, I mean, you said you were there with FDA. So are they thinking about addressing this with new guidelines, environmental metrics? And that's, what's going to, in cell therapy, let's be real clear. I mean, like you just said, I mean, we've stumbled a couple. Now, let's be clear. We're talking about cell therapy. So are we seeing that there's going to be a plethora of guidance? Is there going to be new guidelines? I mean, obviously, we didn't know what we didn't know, which means now we're going to have a guidance, right? I mean, that's usually the way it works. So.
Yes, for those of you who weren't aware, about a year and a half ago, I don't know the exact timing, but there was a grant that was put forth to the Standards Committee body, which is basically an industry-driven organization. Everybody sitting here in this audience can participate. You can look online at FDA SCB. Nexite, I believe, is the group that was actually contracted to gather all this information. They are looking at all parts of cell and gene therapy, from the infrastructure, air, facilities, and as you see, there's a lot of these new modular systems, which I think can improve, but also reduce the cost involved. And I've actually heard the FDA start to say that they really are kind of lending themselves in that direction. And so you've got those pieces, but you also have data standards, you also have QC and method standards. There are every part of the infrastructure around CMC that is being looked at. And so yes, it's a mandate put forth by the FDA. They have spent money, Congress has asked them to do that, and so there is an actual concerted effort going on.
All right, let's do another reality check, and that was obviously, look at the crowd. I mean, considerable interest, right? But let's talk about the reality. There's 2 approved out there. So Peter, I mean, they got to be facing a headwind. I mean, they got to be fighting it too, right? So what I'm saying, we all think it's going to be a slam dunk. I don't hear it yet,
Being a pioneer is not easy.
Right.
I think if you ask any of the folks who worked at Dendreon, right, with that first immunotherapy for prostate cancer, there were a lot of challenges and headwinds there. A lot of lessons learned that you can apply moving forward. Specifically for Yescarta and Kymriah, some of the challenges are really on the manufacturing side, but also on the reimbursement side. So how do you, for a high-value therapy in a hospital's budget or a government payer's budget, how do you create a framework that works and creates value for them and they're able to afford paying for the drug? So that was something that, to be quite honest, probably could have benefited from a little bit more of a ramp in being able to prep kind of the different groups who are involved. From a manufacturing perspective, there have been headlines with respect to manufacturing challenges, specifically cell viability. And that one's an interesting one because there was one product for 2 indications, and essentially you had a specification for the first indication, and then you got approval for a second indication, and that spec hadn't necessarily been adjusted, right? And it hadn't been adjusted because there wasn't enough data. And so what ended up happening was, in a commercial environment, you were having a lot of manufacturing failures. And it was actually to the point where it's very noteworthy because it's on quarterly earnings calls where, you know, the leader of that unit has to answer for what's going on in respect to these manufacturing failures. So again, I think there's a lot of lessons from what we've seen recently with those two products. And all of the products that are coming behind are trying to learn from those lessons, apply that moving forward to how they would do something different in bringing their products to market.
So Boro, or either one, I'd like to get both your input on this. So what do you think?
Yeah, I just want to come back to the point on clean rooms. So it's not just about the clean room space, it's about the devices that you actually use. So some devices are fully enclosed, right? And they don't require such a high clean room environment. Our device, the Prodigy, doesn't require that. And we have clinical trials where clinical investigators are running the Prodigy, creating clinical product in a normal laboratory. The laboratory is ISO 7 qualified, right? But because it's an enclosed system, it's like a clean room unto itself. So this is the new way. Beyond centralized manufacturing, there's point of care. We're enabling clinical sites to manufacture their own CAR T cells. And beyond large clean rooms, have hospitals make it in say an ISO 7 or even a Class D environment, and that will dramatically reduce the cost.
Ali?
My optimistic view at the number of 2 approved application, this is really the tip of the iceberg. I think we're basically scratching the surface, and a lot of products that are either in pivotal trials or late phases. And if you think 2 years ago we had zero, other than some niche applications to a handful of patients that cost $1 million or $1.5 million. 2 years ago we have 0. Today we have 2. Tomorrow maybe we'll have 5. And if you look at any industry, similar industry like the monoclonal antibodies and other biologics, if you look at the trajectory, we're on the same trajectory. Despite the fact that most of our products are one-to-one, and it takes 17, 20, 25 days to manufacture, and they cost a lot of money, we're at the same trajectory. We're using closed systems like Boro alluded to. We're using better air systems like Katie alluded to, and we're reducing the cost. And maybe 10 years from now, you and I will meet for a cup of coffee or a drink and say, remember that discussion we had 10 years ago. So I think we're just starting here. And again, I'm optimistic. I'm not sure about others, but we'll get there.
Okay. So we got a lot of folks sitting here waiting to get in the game. So what are the game changers going to be coming up? So Peter, why don't we start with you and we'll do the same thing. I mean, what process improvement, what things are coming up? You know, if I'm going to be number 3. I'm not, you know, I came from a company that was happy to be a fast second. Now we're talking fast third, fast fourth. By then we should have figured it out, or what is coming up? What do you see coming in to be a game changer?
Yeah, so one that's in place right now for a number of oncology products, but not yet on the rare disease side, is what we call cryopreservation of the apheresis incoming material. And essentially what it is, it's a double freeze throughout the process. And again, I'm going to reference Dendreon, because at Dendreon, that process had an 18-hour shelf life for the apheresis going to the manufacturing plant, and then for the final drug product going back to the patient, another 18 hours. Many companies were able to innovate and learn that lesson on the drug product and freeze at the end of manufacturing. So that's almost a common practice for a lot of products. For SGD products, so severe genetic disorder products, we haven't been able to do that double freeze on the front end. And that double freeze is critical because it allows us to decouple the apheresis activity from the manufacturing slot and be able to plan to a more thought out utilization of your manufacturing capacity. So that's a key game changer in the space that I operate in the severe genetic rare disease disorder space.
Okay. We're going to have to start to move it along because we're getting down, believe it or not, we're getting down to the end here. So game changers.
Game changers, automation and closed system manufacturing. So an average GMP process is 2 technicians with paper or electronic batch records, and everything is manual. And there's 6-hour unit operation today, another 4 hours tomorrow, another 7 hours at whatever day, and at the harvest and all that. But if you have a one-stop solution system where you can get your starting material, whether it's frozen apheresis or fresh apheresis, put it in there, put whatever culture media and antibodies, and come back, use computers, try to control that. And it's not far-fetched, because there are Miltenyi and other companies have similar devices that can do that. But I think more closed system, more automation, to me, is going to be the game changer in our industry.
Yeah, I agree with
Boro?
that, and I would also add fresh in and fresh out. When you have a point of care manufacturing at the hospitals, you can take the cells fresh, manufacture locally, and freshly infused, and that's what we've done, and we've shown actually that fresh product is superior than frozen product, because you don't have to freeze, they don't have to, get back and going again in order to have their effect. So I think point of care, automation, fresh in and fresh out.
Okay, in view of that, what I'm going to do is, considering, I think it would be more expeditious for you guys. Think about a take-home message now. All right, I'm giving you a little bit of time while I queue them up. And the rest of you, what I would strongly suggest is, because we're going to run out of time, if you got questions, I strongly suggest you, shall we say, politely incarcerate these people on the way out. So, take-home messages, and I'm going to have to cut it short, because we got another session coming up. Is that okay? Does that work? Okay. So, take-home message. Who wants to take the first shot? You want to do it, Boro?
Automation and point of care. I think that's the take-home message for me.
Take-home message for me is that there's actually a war on talent in this industry. It's actually, I think, one of the key hurdles that we face. And so individuals that may be sitting here who are, let's say, maybe in small molecule or in recombinant protein therapeutics, I've had many folks approach me and say, how do I break into the industry? And I think it's really just actively going out there and selling your previous experience. But I think the time is now to do that, just because this industry definitely has a need for talent, especially in the CMC space.
Right.
So I would say, as we said, we're at the very beginning of this, but with many years foundation. Build and go forward with what we know works and what we know doesn't work. Whether it be a closed system or an open environment, there's so many points of contact with the living cell. I mean, we did everything possible to work within a closed environment and still had a lot of open points of contact where our cells were exposed to the environment. So I would say build on what we know, expand regulations, work with some of the groups that Heidi was recommending, and work together to develop the most optimal metrics possible.
I would say, in addition to those great take-home messages, is everything about these products is moving very quickly, thankfully, for patients. However, what it means is that we have Google and Amazon and others out there doing the big data analysis. Well, we're in it now. We have no choice. So I think we're looking to adopt and use those tools that are being used by other industries. Power industries have used them for years, by the way. They have lots of data points. But we're now looking at it and looking at the safety of the patients and improving the CMC and infrastructure, and we need to take all of that data and really think about what it is that we know about our products and what we need to still learn, and then apply it to the standards and other things that will make these products better and safer.
Ali?
No fear. Pull ahead with what we got, build on what we have. As everybody said, really, I don't have much else to say other than just continue to have better air environments, better closed systems, better automation, faster manufacture, and oftentimes these processes take 15 to 20 days. I think there is a lot of, the field is new and we don't understand everything about it, but we could use the data from many of our colleagues to try to define the best conditions for a CMC or manufacturing process that we could all use and utilize in the manufacturing of these therapies.
Okay, so my recommendation is pretty simple. All right, I got 30 years big pharma and 15 years as a consultant. And usually when people call me, they go around about. Bottom line is, and this is the message, my take-home message for you. I've already stepped on the landmine, okay? Ask me the question so I educate you so you don't. Thank you very much.